Recall Hammerling's work with acetobularia. He was studying the role of the nucleus in regeneration. He argued that the nucleus produces some kind of substance that diffuses up into the stalk and has the power of regeneration. We have concluded from later research that these nuclear materials are information molecules called messenger RNA. If messenger RNA molecules degrade over time, how are they made again at a later time? There must be permanent recipes in the nucleus. We will see that inside the nucleus are molecules of DNA. It is DNA that stores permanently the recipes for building proteins. Here we see chromosomes from the Drosophila fruit fly larva, specifically salivary gland cells. They have been stained, and the individual bands that you see along the length of the chromosome are genes. What happens to chromosomes during the cell cycle can be illustrated with the onion root tip. This is the tip of the root of an onion. As the root grows longer, it does so because cells are dividing. Now, as we magnify and stain the cells, we can see the brick-like cells, and inside each cell is the nucleus, stained here blue. The fact that the nuclear membrane is visible here means the cell is in interphase. The cell is building proteins, it is burning sugar for energy, and doing the necessary activities to maintain life. So if you look at most of the cells in the root tip, they are in this phase of the cell cycle, interphase. No chromosomes are visible. The nuclear membrane is intact. But a fraction of cells are in another phase of the cell cycle, called mitosis. During mitosis, the chromosomes of the cell are visible. The nuclear membrane has disappeared, and it is in this stage that we can begin to see what's happening to these chromosomes. In this picture, several of the cells can be seen with the colored bodies, or chromosomes, visible. Other cells have the nuclear membrane intact, so they would be in interphase. If a cell has chromosomes visible, it is in one of the stages of mitosis. With chromosomes visible, Walter Fleming was able to count the number of chromosomes inside a cell, and he noticed something important: that a cell with 10 chromosomes could divide into 2 daughter cells, each with 10 chromosomes. This suggested that at some point in the life cycle of a cell, the chromosomes were being duplicated. And that immediately suggested that one of the purposes of this chromosomal dance that we now call mitosis was to deliver duplicated chromosomes to daughter cells. In this professional picture, scientists used fluorescent chemicals to stain cells. And most of the cells you see here have the oval nucleus and so are in interphase, but a couple of them are in one or another stage of mitosis. Here we see a scanning electron microscope picture of a chromosome. In fact, they're duplicated chromosomes, still attached at one location along their length. We will learn that chromosomes are really long strands of DNA wrapped around proteins. And a chromosome can be duplicated when DNA itself is duplicated. And the DNA duplication occurs during the interphase stage of the cell cycle. So Walter Fleming noticed chromosomes in various arrangements within the cell. He made careful drawings of them, and from these drawings he was able to put together the possible sequence of events happening within the cell. The first stage of mitosis we will call the condensing stage. Duplicated chromosomes will begin to coil up and condense. The DNA wraps around packaging proteins. In addition, the nuclear membrane breaks down. You see the system of microtubules beginning to separate to the poles of the cell. These microtubules will begin to randomly explore the cell and make contact with the duplicated chromosomes for the purpose of aligning them at the center of the cell. The cell on the left is still in the condensing stage. The chromosomes are condensed, but they still have the shape of the nucleus. The cell on the right is the next stage of mitosis, where duplicated chromosomes are aligned at the center of the cell. This occurs because of the activity of microtubules. They can get longer or shorter, and as they do so, they are randomly exploring the interior of the cell. When they encounter a duplicated chromosome, the microtubule is stabilized. In time, the microtubules and the chromosomes arrange themselves so that the duplicated chromosomes are lined up at the center of the cell. Here we see several cells in the line-up stage of mitosis. The third stage of mitosis is the separation stage. The duplicated chromosomes separate and move to opposite sides of the cell. This, of course, will ultimately result in two cells with identical sets of chromosomes. Here we see some pictures of cells in the separation stage of mitosis. The final stage of mitosis is the division stage. When the chromosomes are separated to each side of the cell, the membrane pinches in, separating two cells, and a nuclear membrane is reconstructed to house the chromosomes in each cell. Here we see some pictures of cells in the separation stage of mitosis. In summary, then, mitosis consists of four stages: the condensing stage, line-up, separation, and division stage. In this picture, we see, again using fluorescent microscopy, the process of mitosis, starting with the whole cell in the bottom left and going clockwise around through the condensing, line-up, separation, and division stage. Mitosis in animal cells allows us to see a little bit more easily the microtubules that are involved in chromosome separation. In this picture, the chromosomes are blue and the microtubules are greenish yellow. In this picture, again, the chromosomes are blue and the microtubules are arranged at the two poles of the cell and spread out to make contact with the chromosomes. Historically, the original idea of how this happened was that the chromosomes are attached to the microtubules and the microtubules pull the duplicated chromosomes apart. We will call this the pulling hypothesis. Notice that the chromosomes are attached directly to the microtubules. So separation of chromosomes occurs when microtubules are pulling the duplicated chromosomes apart. But more recent research revealed the existence of a new class of proteins. We'll call them molecular motors. These molecular motors can attach themselves to microtubules and carry a cargo. This raised a second possibility for how chromosomes are separated. Perhaps the chromosomes are driven along microtubules. So with two rival hypotheses, the pulling hypothesis and the driving hypothesis, it became important to do an experiment to test these rival hypotheses. One suggestion might be to add a drug that interrupts the microtubules themselves, that prevents them from assembling into the rigid rods. But this would not be a good experiment because to destroy the microtubules would be to interfere with both hypotheses. Both hypotheses would be predicting the same result, that chromosomes would not move at all. Another approach would be to only interfere with one of the hypotheses and not the other. And scientists did this using lasers. They targeted one side of the cell and they caused a break in the microtubules on one side of the cell. This would prevent any pulling, but it would still permit driving up until the point of the break. The actual results supported the driving hypothesis. Chromosomes continued to move up until the cut, and then they stopped. The chromosomes should not have moved at all if they were only being pulled. However, this experiment does not rule out the possibility that pulling is also happening. It's possible the chromosomes are pulled and driven at the same time. Much like in a mall, when you walk up the up escalator, you're being pulled, but you're also walking at the same time. Mitosis is incredibly important for the life of all animals. All animals begin as a single fertilized cell. It is when the fertilized cell goes through mitosis that more cells are produced to construct the body of an animal. Mitosis delivers identical sets of chromosomes to each daughter cell, so each cell has the full set of recipes to build proteins.